Okay, so, oh, and we're being recorded. If anyone doesn't want to be recorded, then switch your camera off or leave. So next up, we've got Sepehr Ehsani on principled mechanistic explanations in biology. Take it away.
Thanks very much, Michael. So can everyone hear my voice well? Okay, great. Thank you. So the paper that I wanted to talk about this morning concerns a form of explanation called a principled mechanistic form of explanation, using the case of current challenges in Alzheimer's disease. So I'll start with a very quick introduction on Alzheimer's and then describe the various features of the principled mechanistic explanation. So Alzheimer's disease, or AD for short, it goes without saying that it poses a huge burden on various communities at present, and by 2050, it's actually projected to affect more than 130 million individuals. Modern research on AD can be traced back to the past 50 years, and especially since the early 1990s, there has been a tremendous focus on AD investigations. In terms of the biology of AD, there are two main pathological hallmarks. One of them is called amyloid plaque, that is an aggregate of a protein or a protein fragment, or actually a peptide called an amyloid beta or A beta peptide. These form mostly outside the affected neurons. The second hallmark is a neurofibrillary tangle that is composed of the tau protein. These occur mostly inside the affected neurons. Usually the first hallmark is referred to or is used in the AD field for an explanation called the amyloid cascade hypothesis. But for simplicity, and this is not technically 100% correct, but I'll refer to both these hallmarks as the amyloid explanation of AD. There are other important genetic risk factors for AD as well. One of them in particular is called ApoE. This gene is implicated mostly in late onset sporadic AD, but my point is not to be exhaustive in terms of all facets of AD. So I won't be referring to the ApoE arm of the AD explanation. This is quite timely. I'm not sure if any of you have seen these commentaries over the past couple of days. These concern a treatment being worked on by the biotech company Biogen that in fact concerns the amyloid explanation of AD, and it has been quite challenging. And these opinion pieces refer to the current challenge facing this, but I won't go into any detail at the moment. In 1989, this was the state-of-the-art understanding of the amyloid cascade explanation in AD. At that time, a simplistic model. Fast forward 30 years from that time, 2019, and this is the current state of the explanation. As I mentioned, here we are looking at a neuron. This is the plasma membrane of the neuron, inside the neuron and outside the neuron. These other cells are shown at a larger scale. We have a neuron, another neuron actually, and some other immune component or immune cells of the brain, astrocytes, microglia. So the current idea or the current thinking concerning the amyloid explanation is that on the neuronal membrane, we have a protein called APP. This protein gets cleaved by various enzymes, leading to the generation of that beta peptide I mentioned a couple of slides back. This peptide, the monomers of it, form oligomers. The oligomers go on to form amyloid plaques. In the past, the amyloid plaques were considered to be the toxic species. At the moment, the thinking is more that the oligomers might be the toxic form of the A-beta peptide. But the point is that outside neurons, we have the accumulation of these amyloid plaques, and these lead to neuronal death, various other downstream effects as well. Inside neurons, that evidently this doesn't concern the APP protein. We have the tau protein, these blue circles. These tau proteins, they get a modification that's called a hyperphosphorylation modification. They aggregate, they form neurofibrillary tangles. So these are happening inside neurons, and these two lead to neuronal death. Much of the focus in AD research and also the clinical trials have been on the extracellular amyloid plaque arm of the explanation, even though the tau form is also quite important. In fact, some of the biomarkers being worked on at the moment for AD concern tau. So what's the current problem with AD research? Well, concerning AD in general, there have been upwards of 200 clinical trials over the past several decades. Concerning the amyloid explanation itself, there have been more than 25 clinical trials done, and unfortunately, none of them has panned out. And we have to bear in mind that for a single trial to come to the point that there is enough justification for it to go forward, there has to be enough laboratory and other kinds of evidence for the trial to even begin. 25 is quite a large number. Without going into too much detail, because I want to focus more on the positive account, my argument in the paper is that this amyloid explanation, the model, the mechanistic model, and the resultant mechanistic explanation appear to be fundamentally incomplete. And this incompleteness has, from what I can gather, very little or if anything to do with a problem of translating the medical biological understanding to the clinic. So this is not a bench to bedside issue. In fact, all the predictions of the explanation are being carried through all the way to the clinic. So it's not a translation issue. It's also very difficult to imagine an entirely different mechanistic explanation that would circumvent the amyloid explanation. Even if we could imagine them, and there's no limit to what perhaps we can imagine. There's a limit, but of course the scope is quite wide. If we are to make an impact, and if we are to be pragmatic about what we are offering as a solution, the solution has to engage at some point with what is currently happening in AD research. Otherwise, it would just be abstract. There is nothing wrong with being completely abstract, but if we are to make an impact, it has to connect with, the solution has to connect with current practice and then maybe augment it in some way. So in the philosophy of biology, the new mechanist tradition, again, I won't go into much detail. The full paper has a discussion on this. My argument is that the existing new mechanistic accounts, to the extent that they go beyond reflecting, and that's the key word, beyond reflecting current biological practice, may not be sufficiently critical of it to help resolve the problem I just set out for AD. My framework, I'm calling a principled mechanism framework, is a model of explanation that supplements or augments mechanistic elements in an explanation with nomological principles. And these principles can be thought of as non-accidental generalizations that may not reach the level of a law claim. So they're not there yet. These principles that I'm suggesting should be hypothesized and then augment mechanistic elements of the explanation. We could call them biological principles. They are operative at the level of individual cells. And they're not laws yet. They could become laws, but at the moment they are based on regularities, on patterns that someone might think, a biologist, a philosopher of a biologist might think, underwrite or govern various various aspects of the operation of a cell. And eventually one or more principles could be formalized into a law, but a principle might turn out to be an accidental regularity, and then it would be thrown out. So it's a stepping stone to have a law. PM, this principle, the mechanism framework, on the mechanistic side, it aims to systematize the mechanistic elements of the explanation. On the non-mechanistic side, as I just mentioned, these biological principles can do two things. They could either inform the mechanistic elements of the explanation, whilst at the same time doing independent explanatory work. They could do either, or they could do both, depending on what the explanation is, what the mechanistic elements are, what the proposed principle is, and so on. There's more to say about the principle, but I'll mention that in the next couple of slides. So let me just quickly mention the mechanistic component of PM. The mechanistic component uses various learnings from the new mechanist literature, various philosophy of mechanism lessons going back to the Cartesians, to have a series of basically pragmatic tests that are supposed to test for what a paradigmatic good mechanistic explanation would entail. So it's looking for good making features of a mechanistic explanation. And why is having these tests important? Because when we systematize it this way, we can help to prioritize which elements of the mechanism we are explaining might be underexplored, might be missing, and so on. And some of these tests, regardless of whether the explanation that we are testing against, whether it fails or passes each one of these tests can also hint at one or more principles that could be posited related to those elements in each test. So just briefly, these are the outlines of what the 5 tests in PM entail. They're not meant to be nested necessarily, one coming after and the next, but it would make sense, for example, for one perhaps to follow the one prior to it. So, for example, we would obviously aim to have the phenomenon we are explaining to be as unambiguously stated as possible. That seems to be a truism, in fact. Test 2 looks for the context in which the mechanism we are explaining is situated, being composed of parts that could be, in principle, detected with various measurement tools in the lab. Test 3 looks for whether all the parts in the explanation can interact with each other in the explanation, or whether they're missing interaction pieces. Test 4 looks for whether in the explanation we can account for one or more important parts exerting change on another part through intermediate means, and whether those changes, test 5, come in a sequence or whether they're disconnected and so on. I won't go into any detail here, but in terms of the AD amyloid explanation, just take test 3 that looks for interactions. Going back to this figure, recall that the amyloid plaque is one hallmark of the disease. The neurofibrillary tangles are also another. But it's not exactly clear how these two arms of the mechanistic explanation, the extracellular and the intracellular, interact. So people might know this, it might be in the background, but having it in a systematic way that the interaction is missing, I think is quite important. And these tests are meant to highlight these in as I said, a systematic way. What about the nomological now component of the PM explanation? What I'm proposing shouldn't come as a surprise, for example, to a chemist, because in organic chemistry, for example, if we want to explain a reaction mechanism, what we would do is what a chemist would do is they would first posit the chain of electron bonds the electron pairs that maybe form a certain bond with arrows, in any organic chemistry course. That's one of the first difficult perhaps things that someone taking a chemistry course would have to grapple with. But they don't stop there. They also augment their explanation with thermodynamic principles. Maybe not in a manifestly thermodynamic way, but they all use, for example, delta G, Gibbs free energy, that is derived from thermodynamic laws, to say whether the reaction that they are going through all the trouble of drawing out mechanisms for, whether it in fact happens or not. What is its yield? So they use both components. In biology, what we have is the molecular reaction mechanism in a way. In AD, for example, maybe it's not as detailed or systematized as it is in chemistry, but we have it. What is missing is the biological equivalent of the thermodynamic principles. What I should also note here is, of course, in biology and cell biology, we have evolutionary laws, we have laws from ecology, we have laws from geography, and they are important. But in chemistry too, we have laws of quantum physics. They don't apply directly. We have to have molecular orbital theory. We can't use atomic orbital theory directly in chemistry. We have to use a chemistry-related law. And there is a gap, perhaps. They might not be reducible to each other, but we have to have a chemistry-specific variation of the atomic orbital theory in the form of a molecular orbital theory. In cell biology, we have population-level laws from evolution, from ecology, but we don't have, in a manifest way, laws that are specific to and operative at the individual cell level. And that is what a biological principle entails. Let me just give a quick example of what a biological principle might look like. This is just a hypothesis, but this is an example of what a biological principle might be. Let's call this the principle of cellular synchrony. Recall that the cell membrane is what separates outside from the inside of the cell. It has been observed for many years, or noted, that the cell membrane vibrates at a specific frequency in all cells. eukaryotes, prokaryotes. It just has to do with the chemistry of the phospholipid molecules in the membrane. That frequency is 10 to the negative 12 seconds. We could propose, based on various observations, I won't go into any detail, that all processes within a cell, the activity of proteins, intracellular transport, and so on, happen in a synchronized fashion at a frequency of 10 to the negative 12 seconds. It follows from this that if this synchronized manner in which these intracellular processes are taking place is somehow disrupted, this could lead to cell death. So in a way, the cell membrane might be acting as a pacemaker for what's happening inside the cell. And I should note this has nothing to do with the pacemaker of the heart, the circadian clock in a physiological organism. This is 1/1,000,000th of a second. This is a time regime that is orders of magnitude quicker than what's happening at an organism level. So even though I'm calling this a cellular pacemaker, again, recall, this is operative at an individual cell level. So if one posits this biological principle, what does that have to do with the AD explanation? Well, it could follow from this principle that perhaps in neurons in the brain, for example, a part of the cortex, the entorhinal cortex, where it's thought that AD starts, if cells are perhaps experiencing a problem with the plasma membrane, perhaps we have asynchrony amongst neurons, and this could lead to neuronal death in that part of the cortex. We could state this without having anything to do with the mechanistic explanation of AD. This in and of itself could become a research question. It could be wrong, but it's just a question that hasn't been posited yet. And that's the point of this, that it can lead to questions that can be investigated. It's empirical. And so this is how a principle could perhaps provide an independent explanation of the phenomena. Mechanistically, too, it can provide clues. So going back to test 3, recall that I said the amyloid plaque and the tangles seem to be disconnected. If the plasma membrane is involved as a pacemaker in the cell, that could be the connection. We could, for example, say that the plaque could interfere with the vibrations of the cell membrane, perhaps leading to neurofibrillary tangle formation, or the other way around. What this biological principle does is it suggests that, and in fact, APP is also on the cell membrane, it magnifies the focus on the cell membrane as an important missing piece in the puzzle as to how these two arms of the AD amyloid mechanistic explanation ought to be connected to each other. There is much more to say, but perhaps what I'll just end this on is to say that a chemist would find the PM framework to be quite in line with how organic chemists approach explanations, I think. But even a physicist would find analogies. Just explaining, the age-old clock mechanism. If we are to explain how a pendulum clock operates, do we just state the activities and parts of the pendulum clock and that's it? No, one of the main important parts of just explaining a simple clock that we think we are all done with is to appeal to principles of gravity, to say how the pendulum works, or principles having to do with friction as to how one cog wheel exerts its movement on the next. So having a mechanistic component and a nomological component to the explanation should be, I think, a natural extension of what happens in physics, what happens in chemistry, to extend it to cellular biology. The full paper is available here in case you'd like to take a look at a bit more detail, and I'll be very happy to discuss any questions that there might be. Thank you. Thank you so much.